So the rest of this chapter, the rest of this lecture is going to be spent on eukaryotic cell, really focusing in on that. And the first part is also applicable to the prokaryotic cells, and that is that cell membranes and the details of cell membranes and how they are the gatekeepers. Again, not the wall, not the cell wall, it's the plasma membrane or cell membrane. It lets some things in, but not everybody in. Here you see these guards. If you don't have a pass, you cannot go in to see the queen. I'm sorry, it's just not going to happen. That's what the plasma membrane does. So, little hint from before about this term phospholipid when we were talking about the endosymbiosis idea. And so we're going to get into a little bit of the chemistry of the phospholipid. And phospholipids are made up of two regions. This is the main molecule that makes up the plasma membrane. And this region here is called the head of the molecule. And this region here are called the tails of the molecule, of the phospholipid molecule. This head region is said to be hydrophilic. And literally hydrophilic means water-loving. So this region of the molecule really orients towards water. It's attracted to water. And so important here for the name of it is it actually contains phosphorus. So of the phospholipid, this is the region that contains phosphorus, and it is named after, the molecule is named after both the head, the phosphorus-containing head, and the lipid-containing tails. So lipids are like fats, and as you probably know, oil and water don't mix, and oil is a fat. So this region of the molecule is hydrophobic meaning fearing water. The hydrophilic head is also known as polar, that water is polar. Hydrophobic is nonpolar. That means that it is not attracted to water. In this tail are lots of carbons and hydrogens, just like most fats. So you've got this structure that is one of the main, most abundant molecules in the plasma membrane, called a phospholipid, and it's got two parts, two regions, one that's attracted to water and one that's not attracted to water. And this has a definite effect on how the plasma membrane looks. So what it forms is something called the phospholipid bilayer. Bi means two. So what happens is that the heads of the phospholipid are attracted to the surrounding water. And there's water inside and outside. So the heads are all kind of oriented out or in towards the center. And what happens to the tails is that they're just kind of attracted to each other. So they form these little structures in here, these little regions of attraction in here. And so we get this bilayer of phospholipids. So that's why we call the plasma membrane or the cell membrane, we call it the phospholipid bilayer, two layers. There's a nice video on Blackboard about this if you want some more details. So this is the main molecule of the plasma membrane, but it is not the only molecule found in the plasma membrane. And we're going to delve into looking at some of those other molecules, including all of them categorized. We got our phospholipids, right? That's absolutely there. We have this molecule called cholesterol. Cholesterol has a bad reputation. People can have too much cholesterol floating around in their bloodstreams. That is absolutely true. If you have too much cholesterol floating around in your bloodstream, you can have blockages in your arteries, you can lead to heart attacks and all these other bad things. But cholesterol inside the plasma membrane, and you see it here, cholesterol is another type of lipid that, remember the tails are lipids here. So notice how cholesterol is hanging out with the hydrophobic nonpolar tails. That's what they do. And what cholesterol does for the cell itself, and again, the only lipids that this is pointing to are cholesterol molecules, is that they help keep that plasma membrane flexible. When it's too hot out, that those cholesterol molecules are going to keep the plasma membrane sticky. And when it's really cold out, it's going to allow the tails of the phospholipids to kind of slide around a little bit more. So it helps with flexibility. Next that we're going to look at are these short chains of carbohydrates. And carbohydrates are almost always depicted as hexagon shapes. So it's a string of carbohydrates, and they often form this Y shape right here. And notice that they're attached to some other molecules. So these short branched carbohydrate chains are acting as fingerprints for your body. Just like your fingerprints on your hands are unique to yourself, these carbohydrates are kind of unique to your body. Not quite like regular fingerprints, but basically what they allow your cells to do is to recognize itself. So if your immune system is trying to attack foreign objects like foreign bacteria or something else that shouldn't be in your cell, inside your body, these carbohydrates say, hey, don't attack me because I'm actually you. So recognition, fingerprints. You can have these carbohydrates attached either to proteins or to the lipids, the phospholipids. Either one, it works. And then finally, there are just a whole heck of a lot of proteins in the plasma membrane. We're going to go into some details about the different kinds of proteins that you might have in the plasma membrane, but this one right here we say is a transmembrane protein because it goes all the way through, so transmitting, going through the entire phospholipid bilayer. There are some that are just surface proteins that float on the outside or attached kind of to the outside. And that can be on the inside of the cell here or the outside of the cell here, but they're just hanging out on the surface. So those are just a heads up on the different proteins. We're going to, right now, go into some detail on what the jobs of some of those proteins are. Just remember, we're oriented here. We've got a phospholipid bilayer. There's our cholesterol. There's our carbohydrate chains that act as fingerprints. And cholesterol there. But let's take a look at the purple guys, because those are our proteins. And for the most part, the ones that we're going to spend the most time thinking about are transmembrane proteins. So three of these are always transmembrane proteins, and these ones over here can be either surface or transmembrane. So the first one that we're going to talk about is the receptor protein. And receptor proteins, they, just like a wide receiver in football, right? They receive molecules of information. And what this does is this molecule right here goes inside the receptor protein. The receptor protein grabs onto it and then sends a signal into the cell, hey, I've got this thing attached to me now. You need to change what you're doing. It's like a shout out to the inside of the cell. Hey, like a game of operator. Hey, change what you're doing. Figure it out. Respond to this. This is important. The next one is called the recognition protein. And notice the shape of this is kind of Y-shaped. It has a very similar function to these carbohydrate chains. It helps your body tell itself from non-self. So body can recognize itself, body will not attack itself, and therefore you function properly. If you have an autoimmune disease, a lot of times this happens because the recognition proteins aren't able to do their job and the body is hyperactively attacking itself. So the third kind of protein that you can find in the membrane are these transport proteins. Transport proteins actually bring things into the cell. So receptor proteins don't bring things into the cell, transport proteins do. You need lots of stuff in your cell. Remember that the membrane is the gatekeeper, and the gatekeeper doesn't let a lot of things in, but these transport proteins do, especially molecules that you really need. So you're going to be bringing in like glucose or something along those lines. You're going to be bringing in proteins that your body eats and like you ingest and needs to get into the different cells. The final one that we're going to talk about are the enzymatic proteins. Enzymes are any molecule, usually a protein, not always, but usually a protein, that makes a chemical reaction happen faster. And sometimes it's really important that that chemical reaction happen faster at the level of the plasma membrane. So there you go. The four main categories of proteins that you're going to find in the plasma membrane. So I wanted to go into some details about some of these proteins and give you some reasons why they might be interesting to you. So the transport protein, that was the third one that we talked about there. It's the thing that it's the protein that allows things to come in and out of the cell. And so your body needs a lot of things. One of the things that it needs is chloride ions. And so there is a disease called cystic fibrosis that is a problem with the chloride ion transport protein. Basically, that protein is not made properly. And so your body can't allow, if you have cystic fibrosis, it can't allow chloride ions to go in and out of the cell. And since your body needs that to happen, this is actually a fatal disease. It's fairly common. I've known people with cystic fibrosis. All children are tested for cystic fibrosis at some point or another, but about 30,000 people are living in the United States with cystic fibrosis. One of the side effects of cystic fibrosis is that you cannot clear out the mucus from your lungs. And so in this diagram, what you see is this patient with cystic fibrosis having her back thumped or pounded on in order to loosen up the mucus so that they can get rid of that mucus so that the lungs aren't damaged too much. But there are machines that can also do this thumping as well. And so that's helpful. There's these treatments for cystic fibrosis, but at this time there is no cure for cystic fibrosis. If you know somebody or if you have cystic fibrosis yourself, the chances of you living into your 40s are next to none. And that's a really scary thing, and something that we should all know about. So chloride transport protein not functioning. We'll learn more about this in lecture. The next one is the recognition proteins. A lot of people are faced with getting organ transplants, and these recognition proteins can become a difficulty if you are not an exact match for the person who donates an organ to you. So I like the color coding here. This molecular fingerprint, it's all purple inside this person, but this person needs a new liver. And the donor liver is sort of a match to it, but then not exactly a match to it. So what this means is that the person's immune system can attack this liver because they see this bit that's not recognized and they think it's foreign. And so what can happen is that liver can be rejected, and that person could very potentially die from a rejection. The good news is that there are drugs that you can take that suppress your immune system, but they're not picky drugs. If they're suppressing your immune system, they're suppressing your entire immune system. And so you are more likely to get sick from having this transplant. You're more likely to get a cold, or you're more likely to get other bacterial infections. And so if you ever do get a transplant, you have to keep monitoring these things. It's best if you can get a transplant that's an exact match, and family members are much more able to match you because these proteins are coded for by the DNA, and you're more similar to your family members with regard to DNA. So just kind of something to keep in mind. The next protein that we're going to talk about is the receptor protein. This is the first one we talked about on the side where I was introducing proteins to you, but it is a little bit more complicated than the other two, so I kind of saved it for last. We're not going to talk about the enzyme proteins in this chapter. So the receptor proteins are the ones that grab onto other molecules and send that signal. They don't actually send the molecule, but they send the signal into the cell. And a really common example, something that almost everybody's familiar with, is the molecule adrenaline. Your body makes adrenaline in your adrenal glands. And whenever you're under stress or you're excited about something, your body sends out adrenaline, and it sends it out to all the cells in your body, and they have these receptors called beta receptors. And the beta receptor grabs onto the adrenaline and sends that signal on, hey, I've got this adrenaline, guys, we need to step up, we need to make the heartbeat faster, we need to sweat, we need to increase blood pressure, all of these things. And that's great if you're in a scary situation. But when you're in a situation that maybe shouldn't be so scary, maybe you don't want it to be scary, maybe you're giving a speech in front of a class and you start to feel a ton of anxiety and you don't want to feel that anxiety, it can be overwhelming. So we have this molecule that we've come up with called beta blockers. And beta blockers are literally have a little bit of the same shape as the adrenaline, and it fits into the beta receptor, but it doesn't tell the cell to get anxious. It doesn't tell the cell to, oh, we need to have the heartbeat faster or increase blood pressure. It blocks that from happening. And so when you start to get a little anxiety, the adrenaline comes and it can't get on the receptor because the blockers are blocking it. And so you end up, and this is pretty cool, not having the faster heart rate, not having increased blood pressure, not having the sweats, not having that anxiety, because this molecule right here is being blocked by this molecule. So beta blockers, kind of cool. We do the same kind of thing with if you ever take Advil for pain. The pain receptors in your cells are being blocked by the Advil, and so you don't feel the pain anymore. Science and medicine take advantage of this a lot. So we're still talking about the membrane, and we're going to talk about it for a while. It's an important structure. Let's talk about how it acts as a gatekeeper and how things move across the membrane. And there's a couple of different ways that molecules move across the membrane. We'll talk about diffusion. We'll talk about something called passive transport, and passive transport can be either simple or facilitated. We can have active transport, and we can have bulk transport. There's that term endocytosis I've used a couple of times with you already. So let's delve into how things actually do get across the membrane. The first thing that we need to know about is diffusion. And diffusion is a term that can be used whether a membrane is present or not. But in biology, we really focus on proteins. But let's get the diffusion concept down first, and then we'll talk about how things move across that membrane. So diffusion is where you have an area of high concentration, like this dye right here, all the molecules that make up the dye, in something that has less of concentration. In this case, we've just got some plain water. So diffusion is the process by which the molecules that are in high concentration spread out and become evenly distributed across the rest of that fluid. So we have high concentration and then we have even concentration. That's diffusion. I think this just happens. This is just a physics kind of thing. Molecules bump into each other, they spread out. That's diffusion. So keeping in mind diffusion, we're going to talk about passive transport. And this is where we really start thinking about how things move across the membrane. So if you've got a high concentration of molecules outside of the cell and a low concentration of molecules inside the cell, some of those molecules can in fact move directly across the membrane. This is called simple diffusion. It's not simply diffusion. It's not this stuff, which is plain old diffusion. We modify the word diffusion by putting the word simple in front of it. That is the term, simple diffusion. So some molecules can do this, but here's the trick. Not all molecules. They have to be really small. That's the first thing. And the molecules have to be hydrophobic. Remember hydrophobic. Hydrophobic was the term we used to describe the nature of these tails in the phospholipid bilayer. So if a molecule is small and hydrophobic, like we see with this carbon dioxide molecule, it can just diffuse right across that membrane through the process of simple diffusion. If, however, the molecule is bigger, or if that molecule is polar or hydrophilic, it's not going to be able to just kind of wiggle its way through the phospholipids. It's got to go through a transport protein. And sometimes you call them channels or sometimes you call them carriers. It doesn't matter for this class. What I want you to know is that if you have to go through a transport protein, you're doing facilitated diffusion. And facilitate just means to help. So you've got simple diffusion, which doesn't need any helpers because they're small and hydrophobic. But we've got facilitated diffusion that needs a helper, in this case a transport protein, where it moves through. But notice that it's all moving from an area of high concentration to an area of low concentration. And it'll keep doing this until it gets evenly distributed, just like we saw with the dye in this slide here. So high concentration, low concentration, you're going to move down your concentration gradient. And that's true for most things in your cells, but not everything. Sometimes what you really, really need is a high concentration of molecules. And so when you need a high concentration of molecules in a particular area, you're going to fight against the idea of going down your concentration gradient. So we need active transport. And active transport uses transport proteins, but it also requires energy. And so these molecules here, these little molecules here are actually moving up their concentration gradient from an area of low concentration to an area of high concentration. And the only way they can do that is if they're being shoved in there by energy, and that energy is ATP. And ATP is the main form of energy that all of our cells use. So active transport. The good example of this is your stomach is highly acidic, and the only reason it can stay highly acidic is if these ions are being pumped constantly into the opening that is your stomach. So it's going against the concentration gradient. The cells are making the ions and then pumping the ions outside of the cell into that big gaping hole that is your stomach and concentrating them right there. Active transport. We talked about bulk transport as well, and bulk transport is just moving tons of big things in all at once. So, or out all at once. We're going to start with endocytosis. Endo means into, cytosis means the cell, cyto means cell, cis is the process. So endocytosis is the process by which you move big things into the cell. So you can see right here an example of a cell is eating another cell. That would be endocytosis. You can kind of see the invagination that we talked about before. Or it could just be a really large particle or a bunch of those particles. You have the plasma membrane folding in, forming this structure called a vesicle, and that vesicle is brought into the cell. It's now inside the cell. That pinching off of the plasma membrane is very, very common, and this is plasma membrane right here surrounding whatever that was that we wanted to bring in. Endocytosis. The opposite of endocytosis is exocytosis, and with exocytosis, we're talking about how we get rid of from our bodies. We get rid of large quantities of large items. So, for example, maybe you're making a lot of adrenaline in your adrenal glands on top of your kidneys, and you want to exude that adrenaline out of your body, out of your cells and into the rest of your body. You're going to do that through transport vesicles. How they get in the transport vesicles, stand by on this. We'll talk about that eventually in this lecture. Just stand by for now. So the transport vesicle moves over, butts up against the plasma membrane, and then fuses with the plasma membrane. And when that fusing happens, whatever was inside of that vesicle is now shoved to the outside of that cell, now able to move throughout the body. So where did that vesicle come from? That's what we're going to talk about very soon. Heads up, it's the endomembrane system. Just heads up on that. We'll get to that soon. And in fact, I'm going to go ahead and stop this video now so that we can save all of the big parts of the cell, of the eukaryotic cell, and these are the parts that we're going to be talking about That vesicle is formed in this set of structures right here, the endomembrane system. Let's go ahead and step in now and we'll pick it up with the third video.